So going back to these problems that we defined earlier, in the first problem, we asked how it would be possible to measure all 20,000 genes in cells. And this technology that we introduced, single-cell RNA sequencing, is the answer to that. This makes it possible to measure the gene expression for all genes in 1 million cells. And now we'll move to talk about the second problem that we posed, which was whether it would be possible to delete individual genes in cells and see what the effect is on the cells and specifically on the gene expression in those cells. And so this is part C, where we'll be talking about this second technology that focuses on combining these gene perturbations with the cell readout. So going back to the scheme that we saw earlier, so we spoke about how from a population of cells, we can use single-cell RNA-seq, this first technology, to produce this gene expression matrix. And now, in this second technology called Perturb-seq, what you can do is introduce deletion of a specific gene to each of these cells, and then output a similar kind of matrix, but now where you can see how this matrix differs from the matrix that doesn't have any gene deletion or any perturbation. So going back to our example, the adipocyte populations that we have, so we saw that in the single-cell expression matrix, we could identify each of these three populations by each of these three clusters of genes expressed in those clusters of cells specifically. And so how would this look now if we combine this with the perturbations that the second technology permit us to do? So in this case, for example, we might delete the gene SCD. This is a gene that's known to be important for regulation of the lipogenic program, the lipogenic state. And so if we delete this gene SCD, what we'll actually get out is a very similar matrix, except we'll have a row added that shows that this gene SCD was deleted, and we'll have a count to show which cells this gene was deleted in. And so this is showing for one gene, but in theory, we could delete many genes and also different genes in different cells. And so what we might see is upon SCD deletion, given that this is a gene that regulates the lipogenic program, that actually we completely lose expression of these two genes, SREBP1 and FASN, which are key genes of the lipogenic state, and therefore we completely lose this cluster of cell 5 and 6 that we had previously as a result of this deletion. And in this particular case, I picked a gene that we know regulates the lipogenic state or the lipogenic program. But in another case, it might be a gene that we know nothing about, and so this would be an extremely revealing and interesting result. And so to dive in a little bit more to how Perturb-seq actually works, it relies on this gene editing technology, CRISPR-Cas9, which you may have heard about in the news, and it won the Nobel Prize a few years ago. And the way it works is this protein called the Cas9 nuclease is guided to specific parts of the genome, so specific regions of DNA or specific genes. And at that specific location that it's guided to, it will insert a double-stranded break. And so what we can do is we can manipulate which genes the CRISPR-Cas9 system is guided to by providing different guide RNA molecules. So we'll provide a guide RNA molecule for a gene of interest that we are interested in deleting or knocking out. The Cas9 system will be guided there, and then it will lead to this double-stranded break, therefore deleting the function of that gene. And so this is the technology that Perturb-seq relies on. And so to remind you of what we spoke about, Perturb-seq is the combination of two things. On the one hand, using Cas9 to insert specific deletions in individual cells. And as I mentioned, you could have different genes being deleted in different cells. Combining this with a similar readout as we saw before, which is this single-cell RNA-seq matrix, where for a given cell you can count the number of RNA molecules, but combining this with the actual perturbation identity, i.e. the specific gene that was knocked out in that cell, here denoted as A or B. And so what's the readout? There are two readouts that you'll get. One is this expression matrix that we're familiar with, hopefully by now. And the second is this perturbation identity matrix that will tell us for each of the cells in the expression matrix whether there was a perturbation, and specifically what gene was perturbed or what gene was knocked out. So you can see here, for example, that in cell 1 we have the gene expression profile, so the expression levels for every gene in the genome in this cell. And we also know that this expression profile is the result of whatever programs were already inside that cell, but then adding the deletion of MLX IPL. And likewise in cell 2, we can see the gene expression profile and the gene CBPA that was knocked out, and so on. Finally, we also have some cells, always in perturb-seq experiments, where we don't knock anything out, so that we can compare each of these perturbation groups to the non-targeting, it's called control. Or in other words, the expectation with no perturbation. So the advantage of this is that we can get this very informative, rich transcriptomic readout, the single-cell expression matrix. We can also, as I mentioned, do these perturbations in a pooled format such that different cells will receive different perturbations, thereby allowing us to cover more of the genome. And we can use this, the combination of the cells without perturbation and the cells with specific perturbations, to infer the functions of these specific genes that are perturbed. There are, however, still some downsides. One is that even in spite of this pooled format that allows us to profile many perturbations in parallel, it's still very time-consuming and would probably take one Tiago doing these experiments 22 years to actually profile cells receiving every single of the 20,000 perturbations in the genome. And so this is one very key motivation for this challenge, which is falling short of being able to experimentally test in the lab every single perturbation. What we would like is to be able to use machine learning to predict perturbations, their effects, to predict this matrix, the effects of the perturbations on individual cells, and hopefully highlight perturbations that we predict to be particularly important in the context of adipocyte biology and obesity. And so we'll be talking more about that in the final lecture on data and the crunches that you'll be doing.